Thank you for stopping by to listen to me talk about genetic diversity maintained in comparison of captive propagated and wild populations of Lampsilis fasciola and Ptychobranchus fasciolaris. Given the decline of Unionids, propagation has been identified as a viable conservation tool for this taxa, and many mollusk recovery plans include some kind of party strategy, where party is propagation, augmentation, reintroduction, translocation, and introduction strategies. More than 6 million propagules have been produced, and more than 1 million of those have been released. And success of these efforts is difficult to assess since information is either not widely disseminated or monitoring of genetic diversity has not occurred. And best practices for party strategies aim to maintain genetic diversity and variation of propagules, avoid outbreeding depression, and avoid hatchery selection. And more empirical evidence is needed to validate these guidelines and best practices. We specifically aim to answer these questions. Does genetic diversity differ between captive reared and wild individuals for Lampsilis fasciola and Ptychobranchus fasciolaris? Is there genetic structure between the captive reared and wild individuals for each of those species? And are there any temporal or geographic patterns of genetic diversity and structure in Lampsilis fasciola? So for Lampsilis fasciola, the wavy-rayed lamp mussel, we had 8 microsatellite loci. In our data analysis, we have included genotype data from Galbraith et al., which were collected in 2008. So we have 2 sites on the Grand River and 3 sites on the Thames. We also have individuals from the Grand River in 2018, as well as juveniles that were propagated from 12 females and then exposed to smallmouth bass. So on this map here, this is showing you southern Ontario. So our Grand River individuals came from here, our Thames River individuals came from here, and then the captive propagation occurred at the White Lake Fish Culture Station up here. And this propagation trial occurred before the design of our experiment, so the genetic data for the mothers was not available to us. Here's a summary table of our genetic diversity metrics going from left to right. You have number of individuals, allelic richness, rarefacted allelic richness, which accounts for differences in sample size, effective number of alleles, observed and expected heterozygosity, and an inbreeding coefficient. And all of these values were meaned across all samples and loci. And then we used a Kruskal-Wallis test to detect any significant differences between the groups for all of these genetic diversity metrics. And in those analyses, we did not detect any significant difference between the groups for any of the genetic diversity metrics. Here is a table showing pairwise FST below the diagonal and DEST above the diagonal, where bold values indicate significant comparisons. And looking at this table, we do not see any values that would indicate to us that those populations are differentiated for comparing the Grand River wild individuals to the captive reared individuals. And the global FST for this group is 0.058. And then a structure analysis for these individuals reveals 2 populations, with the Grand and the Thames rivers being distinct. So you can see here that they're 2 separate colors. And then within the Grand River, you can see there is no additional substructure between the propagules and the wild individuals. So this would be one genetic population, and the propagules, which are represented here, are not distinct from the rest of the wild Grand River individuals. And this PCOA, when you can see that same pattern. So in yellow, again, is the Thames, and maroon is the Grand. Hot pink is our juveniles. You can see that there is structure between the Thames and the Grand, but we have complete overlap between the propagated juveniles and their adult wild counterparts. For Ptychobranchus fasciolaris, we had 9 microsatellite loci. We have wild individuals from the Sydenham River collected in 2018, and we have juveniles that were propagated from 7 females exposed to Johnny and black-side darters that were reared at the White Lake Fish Culture Station, which is up here. The propagation trial happened before our study was designed, so we do not have any genetic information for their mothers. This is a summary table of our genetic diversity metrics, just like the one I showed you before. And again, we did Kruskal-Wallis test to test for significant differences between the groups for all genetic diversity metrics. And again, we did not find any evidence of that. For the pairwise FST and DST, we had very, very small numbers close to zero, indicating that these are the same genetic populations. And then PCOA reveals that same pattern, complete overlap between the wild in yellow and the propagated juveniles in maroon. So overall, we see no evidence of significant differences between the genetic diversity or structure of wild and captive-reared individuals in either species. We see no evidence of temporal genetic structure in Lampsilis fasciola between 2008 and 2018. And these temporal and river structure results are similar to other studies on this species, like Galbraith et al. 2015 and Zanata et al. 2007, with structure between the Grand and the Thames rivers. We also see a similar maintenance of genetic diversity in wild and captive-reared individuals and wild individuals in both species, despite lower estimated effective population sizes in the captive-reared group. We do not see any distinction between wild and captive-reared individuals for either species. So this really brings into the question the role of multiple paternity. Multiple paternity is well documented in other Unionidae species and likely occurs in this species. So we have questions about the paternal contribution to each glochidial brood, since our data show that with 7 to 12 females contributing, there is maintenance of genetic diversity. And results from the study are currently in revision at the Canadian Journal of Fisheries and Aquatic Sciences. We wanted to take a deeper look at some of these questions. Specifically, is genetic diversity or structure different between captive-reared and wild Lampsilis fasciola? How many males contribute to a glochidial brood? How much variation is there between glochidial broods? And do microsatellite and SNP-derived genotypes differ in the patterns revealed? We were able to perform a hatchery experiment, and it was really important to us to make sure we had genetic data for the mother, since we did not have that for the first experiment, and that we also knew which juveniles belonged to which mother so that we could do analyses by family group. We collected 12 gravid females from the Grand River. They were brought back to the White Lake Fish Culture Station, where the glochidia were then used to inoculate smallmouth bass, and then those smallmouth bass were kept grouped and separated by family. Once the juveniles metamorphosed and fell off the fish gill, we kept them separate until they were harvested when they reached about 1 millimeter in length. And to answer these questions, we wanted to use a microsatellite and SNP approach. So this graph is a comparison of the number of samples from each gravid mother included in each marker set. You can see that there is variation in the success of propagation. For some mothers, we were only able to collect a few individuals, and for other mothers, like mother 2, we were able to collect about 30 juveniles. For the microsatellite dataset, you'll see that we have a pretty high number of individuals included in the analyses, where for the SNPs, we have reduced representation. And we can do that because of the power of the SNP approach. Here's a summary of what samples we have included in the analyses. We have those same individuals from the first experiment, so we have the Thames River individuals from 2008. We have the Grand River individuals from 2008 and 18. We have our 2016 cohort of juveniles, where we don't know the maternal data. And then we have our 2018 juveniles, which are represented by 12 females, and those 12 females are accounted for in the 2018 Grand River wild population. And just to remind you where those are on the map. I do want to note that this data set was reduced to 7 microsatellite loci due to missing data in the 2018 cohort of juveniles. Once again, we compared these genetic diversity metrics using a Kruskal-Wallis test, and again, we did not detect any significant differences between the groups for any genetic diversity metric. And the global FST in this case was 0.066. So far, we're seeing some maintenance of genetic diversity. Here we have a pairwise FST and DST table of the genetic differentiation, and in this case, the estimated values both are low enough to interpret little to no genetic differentiation between our compared groups. Here we have a structure analysis. This is the Grand River individuals. This is our 2016 cohort of juveniles. This is our 2018 cohort of juveniles grouped by family. Here's our Thames River. So we still see that pattern of the Thames being genetically distinct. This analysis would also indicate that the 2018 cohort is distinct from the other Grand River individuals. And we think that this is actually an artifact of microsatellite marker scoring rather than a signal of our actual data. So we're going to do some additional analyses to look at this deeper and to make sure that this is an actual signal, that they're distinct and not an artifact of microsatellite scoring. We did a PCOA to visualize the genetic differentiation. Here are the wild grand individuals, and the mothers of our 2018 cohort are included in this group. This pink is the 2016 juveniles. This purple color is the 2018 juveniles. And we see that there's quite a bit of overlap, so this would not indicate that these groups are genetically different. I wanted to take a closer look and kind of look at that by family. So now you'll see that the fuchsia color, each symbol is representing a different family of the 2018 juvenile group. So here we have those Grand River wild individuals. Their mothers are included in this data set. Here are the 2016 juveniles, and here are the 2018 juveniles. You'll see that as far as the family goes, there's a lot of overlap, and we do see kind of some separation for these groups along the x-axis, but the variation explained by this axis is only 6.9%. So additional analyses would need to be done to indicate differentiation between these groups. Overall, so far in our analyses, we see no evidence of significant difference in genetic diversity between the wild and the captive-reared Lampsilis fasciola. The pairwise FST and DST values are low, indicating no differentiation between the groups. And PCOAs of genetic distance show overlap between the wild individuals and the captive-reared cohorts, indicating little genetic differentiation. And even though the structure analysis shows more differentiation, we think this is an artifact of microsatellite scoring and sample sizes. So we're going to be doing some additional analyses to investigate that. Our next steps are to complete additional genetic structure analyses, for example, BAPS, parentage analyses to assess male contribution, analyses of genetic diversity, variation by glochidial brood, and genetic diversity and structure analyses with our SNP dataset to compare the results between marker sets. And I'd like to end this by thanking everyone on this slide.